Good morning, ladies and gentlemen. Good morning, ladies and gentlemen. After a great day one, there are a few heads this morning that are a little fuller than most, and some are a little thinner than others. But we're very fortunate to be blessed with a great turnout again this morning. A few new faces in the audience. So today, obviously, for those that know the flavor of day two, we've got the breakout sessions after Dr. Elaine Ingham's presentation this morning. So once again, as I stated yesterday, the only I here is in innovative. It takes a team of individuals, a great group of directors, our executive director, Carrie James, to pull this event off. And without your attendance, our effort is in vain. So thank you to our sponsors that are going to be mentioned later on, and thank you once and again as your conference chairman for attending. Have a great conference and enjoy day two. Without any further ado, our Master of Ceremonies, Archie Wilson.
Thanks, Blake. Good morning, everyone. Good morning, everyone. There we go. We're ready to go. Okay. A couple housekeeping items right off the start here. Checkout time for the hotel is 11 o'clock for those people staying. People that are here for the first time today, welcome. A couple things that I'll make sure that you know today and might be repeating for people from here yesterday. Meal tickets are in your name tag. The yellow evaluation forms, take time to fill those out because, again, that's a big help for us. CCA sign-up sheets in the back corner there, just under the, wave your hand, Paul, so everybody knows where it's at. Thanks. Question cards on the table, so if you have questions of the presenters, please make sure you write them down, hold them up, and we'll collect them at the end of the session. Time permitting, we'll ask the questions. Ask Me ribbons, again, you'll see some directors and other people with a red ribbon on the bottom of their name tag, and for those people, they've got years of experience that we can probably share with you, so don't be afraid to ask them. They've probably run into some of the challenges you're running into yourself. Cell phones, like I said yesterday, I think it's almost getting to the point we don't have to talk about this, but it's just a gentle reminder to everybody, put them on vibrate, put them on silent, so we're not interrupted. Kate, can you put your hand up or stand up? There you go, thank you, Kate. Kate Proctor is our conference reporter, and we appreciate what she's doing here. She might be asking you as a participant or as a speaker to talk to you, so please give her some time if you can. I want to thank our major sponsors, again, being Syngenta and Country Guide. And if you look on your program at the back, there's a bunch of other sponsors that have helped make this go. And as was mentioned a second ago, it's pretty important that we have those sponsors to make this thing work. Without further ado, I'm going to introduce our first speaker of the day, Dr. Elaine Ingham. She's going to speak to us about the impact of fungicide overuse on beneficial soil fungi. This session is sponsored by A&L Canada Laboratories. I'm just used to calling it A&L, so I have to think about this a bit. Elaine is a world leader in soil microbiology and research of the soil food web, pioneering many of the current used biological soil amendment techniques, as well as the testing of soil microbial life as an indicator of soil and plant health. She has been the mentor of numerous soil scientists and practitioners all over the world, helping farmers to grow more resilient crops by understanding and improving their soil life. She's earned her PhD in microbiology with an emphasis on soil from Colorado State University, and I'm not going to say the date because that always ages people. She's got lots of, she's not really that old. She did this when she was 6 in 1985. Very bright lady. She joined the Oregon State University faculty. She was an affiliate professor of sustainable living at the Maharishi University of Management and adjunct. I'm going to have a problem with my tongue today for some reason. She was an adjunct faculty at Southern Cross University in New South Wales from 1999 to 2005. Visiting professor with Melbourne University from 2004 to 2008, and program chair of the Ecological Society of America from 1999 to 2000. She later joined the Rodell Institute as a chief scientist until 2013. You can obviously see here lots of experience in not only our continent, but in other continents. She is currently founder and president and director of the Soil Food Web, where she has developed 3 new methods to rapidly assess soil and foliage-related organisms and is able to assess how soil and foliar biology change with different management practices. She is focused on grower-related issues, the expense of extensive chemical use, and the damage these chemicals inflict on the beneficial organisms in the soil and on the foliage. Obviously, where we see a lot of things changing right now as people look more and more to the biology, this is absolutely a great presenter to come forward here and speak. Please join me in welcoming Dr. Elaine Ingham.
Am I supposed to be down here? My stroll across the stage. So the picture on the screen is the very first picture that was in the calendar about sustainable agriculture put out by the Lexicon of Sustainability. And so I was like the poster child for their very first calendar. And I'm going to expand my talk today, not just focusing on soil fungi, on beneficial soil fungi. I want to do the soil food web. Because fungi don't work by themselves. Just like none of you work by yourself. You have a whole team of people behind you that are helping support what you do. Same thing in the soil. Fungi don't work alone. It's a food web that we need to get back into the soil, and I want to go over that food web with you. So there we go. That was the title that I've been given, but I'm going to expand it. Don't narrow me into just these little topics, because you really need to understand the whole food web if you're going to understand what beneficial soil fungi are all about. So I do want to point out, yes, I've got the alphabet soup after my name, whoopee. And I am Soil Food Web, the president and founder of Soil Food Web Incorporated, and we are a worldwide business. So Soil Food Web at AOL.com, so you can get hold of me if you need to get hold of me. When we start looking at what fungicides do in the real world, yes, they kill some of the disease-causing fungi, but they do a much bigger job on the rest of the fungi present in your soil. A lot of our fungal diseases have become resistant to the fungicides that we use, but our beneficial soil fungi have not become resistant to those fungicides. And so when you use fungicides, quite often what you're actually doing is wiping out the very organisms that will compete with, consume, and destroy your disease-causing fungi. We need to make certain that we get the full food web back into the soil, because it's not just beneficial fungi that attack and inhibit and consume your disease-causing fungi in the soil or on your plant surfaces above ground. It's other organisms in that food web. Do you have those organisms in your soil? And if you don't have those organisms in the soil, we can't call it soil. That's dirt. If you don't have life in your soil, if you do not have the beneficial organisms present, then you're trying to farm using dirt. And you know how successful that is. Not. So we need to get that life back into the soil. And really what I want to explain today is that process of who are all these organisms, what do they do for you, why would you care, and continuing with the theme of the talk. I first got introduced into fungicides as a portion of the food web back at Colorado State University when I was working on my PhD. One of the things we wanted to do, actually it was with my postdoc after I finished my PhD at Colorado State University. I was hired by the Natural Resource Ecology Lab at Colorado State University. And the question that we were trying to figure out is, what precisely do these organisms do out in the real world? Not just fungi, but bacteria and protozoa and nematodes and microarthropods and earthworms. All of those groups of organisms. How many of you have thought about the spiders in your soil? And are they important? Do you need to have them there? Absolutely, you do. And we'll go through all of that food web in hopefully pretty rapid form this morning. I'm not going to be able to talk about everything because I usually give a 5-day course. to talk about really what all we know about biology in the soil. And so I'm taking a 5-day course and paring it down to 1 hour. So I'm going to miss some things, and I hope you'll forgive me if I don't answer all of your questions this morning. My next talk is on compost and compost tea, so I'll be doing a little bit more in the next talk. So understanding what all of these organisms actually do for you in the soil. And the way we approached that question was to take 3 replicated areas in each of irrigated wheat fields on the eastern front range of Colorado, and dryland wheat areas, short grass prairie, tall grass prairie out in Kansas, high mountain meadows in the mountains in the Rockies, and then the old growth ecosystems in that area, lodgepole pine. And we started looking at what is the role and function of these organisms by using different pesticides to wipe out as much as we could of each of the organism groups in the soil to try to determine what it is that these organisms do. And probably one of the first things we really discovered is that there is no pesticide that kills just its target organism group. If you use a fungicide, you're wiping out many more species of the other beneficials in soil than you're affecting of the disease-causing fungi that are present. How do you control, how do you make certain that you're not going to have disease-causing organisms attacking your plants? You need the rest of that food web. You've got to have all these beneficial organisms, because think about how Mother Nature is working out there in the real world. How many of you have walked into an old-growth forest and seen overwhelming disease take out that healthy forest? Doesn't happen. Go to a meadow where we've got good structure, we've got good biology in that soil, and you don't see weeds, you don't see diseases, you don't see pests. So how does Mother Nature do this? So let's be observant and pay attention to how nature is managed to not have these kinds of problems. We don't have a lack of fertility in any of our native natural systems. So let's be observant and figure out what nature is doing. And so I want to talk about that aspect. Do we really need to use toxic materials that are going to be killing way more than the target group? You can't kill just mildew. You're going to kill hundreds of thousands of other species in your soil when you do this, when you use it. That fungicide, excuse me. Just trying to wake you up. So one of the things I did was asked to do back in 1984 when we were working on this project in Colorado, trying to demonstrate what these different organism groups do. We were trying to pick different pesticides. We needed to pick a bactericide, a fungicide, a protozoicide, and a nematicide to take out these organisms in the food web so we could discover what they were important for. So I went to chemical companies and I asked them, can you suggest a fungicide for us to use in this study where that fungicide is just going to kill our fungi present in our soil? Oh yes, absolutely, you should use, well, they always recommended their product. Isn't that strange how they would suggest that their product, their fungicide would be the one that would be best for us to use? Oh yes, we're dealing with business. And I would then ask, well, how many non-target organism effects were there of their fungicide, protozoicide, nematicide? And they would always say, oh, there's absolutely no non-target organism effects. This fungicide only kills fungi, and so our product is the best for you to use. Okay, thank you very much for that information. And then I went into the scientific literature and started searching for papers that told me about, first of all, is that fungicide effective? Will it kill all of the fungi? Will it kill just some of the fungi? And does it have non-target organism effects? And I found literally thousands of papers in the scientific literature, and I summarized them in a review of 12 of the different pesticides that we had been interested in. So that paper was published in 1984, showing all the non-target organism effects of all of these different kinds of pesticides. There is no fungicide that kills just fungi. There are many non-target organism effects. And if you want to look at that literature, go to my website. You can find the reference in crop protection for that paper where we reviewed 12 different common pesticides, so bactericides, fungicides, protozoicides, and nematicides, and herbicides, where it's very clear that you're killing way more than just the target organism with these toxic chemicals. Well, they're toxic chemicals. They can't have an effect on just one species of fungus. So what would we use instead of these materials? So let me, things work. So the fungi, got the right button this time, are going to take those microaggregates and pull those microaggregates with the strands. Fungi grow as long strands through the soil, and they will take things and bind them together and make them macroaggregates. Well, think about what happens when we start building structure within your soil. We're building air passageways. We're building hallways to let oxygen and water and roots move as deep as necessary in your soil. If you're building structure as rapidly as possible within your soil, you don't have to worry about infiltration. You don't have puddling. We're dealing with the compaction. We're opening that back up. So sometimes the organisms may need a little help. The first time you put these organisms back into your dirt, we may have to till one last time to get the organisms as deep as you need them to go. But beyond that, if you maintain that life, you don't need to till. This is the only way to actually do no-till agriculture, is to have these organisms working with you at the direction of your plant, telling these organisms to get out there and do their job of building the structure so that plant can keep oxygen around its root system. If your soil is compacted, if it goes anaerobic, guess what? The disease-causing organisms require reduced oxygen conditions in order to outcompete the good guys, in order to outcompete the beneficials. So you're putting yourself into that requirement to use fungicides if you're allowing your soil to compact. How do you decompact your soil? Grow microorganisms. Get them building that structure for you. You do not want to be killing these guys by tilling, because when you slice and dice and crush and wipe out these organisms as you till and till and till and till, again, you're destroying the beneficial fungi. We're destroying the good guy nematodes. We're destroying the microarthropod. Well, what's their role and function? Okay, we'll get there. Hold on. So building structure, another very important part. Once you've got the microaggregates, the bricks in the structure of your soil house, then we've got to put those bricks into walls. We've got to have windows and doors and roofs and, multi-layer condominiums, and that's what the fungi do. Well, the larger pore structures are going to be made, produced by these higher level organisms. So they have a role and function in structure. You don't have those organisms, you're not going to have the larger pore structures to store your water. As soon as we get structure back into the soil, and yes, you have to have organic matter in your soil because it's not just your plant that will feed these organisms, it's organic matter in your soil. So we need to get that back into our soil to constantly feed these organisms. And then we'll get the higher level predators doing their jobs. But it's not just disease suppression. It's not just building soil structure and getting your roots to grow as deep as they possibly can grow. How many of you grow wheat? Yeah, lots of you. How far down can roots of wheat go? How far down should the roots of your wheat plants go? a minimum of 10 feet. Within a month of that seed going into the ground, the root systems of your wheat crop is entirely capable of being down at 6 feet. By the end of the growing season, that root system should be down at 10 feet. If you're letting your roots grow as deep as they possibly can grow, do you have to worry about water? Do you have to worry about nutrients? You have no lack of fertility. But if you've got a compaction layer in there somewhere, that's the surface of your soil, and here's your compaction layer, the roots of your wheat plant are only going down for maybe 6 inches. And then the anaerobic disease-causing organisms growing in that compacted layer are going to start taking your plant out. Your roots go sideways, and then good luck with fertility because you have put your plants into a coffin, and things grow so well in coffins. So building structure, those things. But one more really important thing is occurring by your plant feeding these organisms and directing these organisms to make the enzymes to go out and pull from the crystalline structure of your sand, your silts, your clays, your rocks, your pebbles, your parent material, boulders, all of that mineral component within your soil. These organisms make the enzymes and the organic acids to pull the nutrients straight from that mineral crystalline form within your soil. There is no soil on this planet that lacks the nutrients to grow plants. There is no soil that lacks the nutrients to grow your plants. Why are you putting on inorganic fertilizers? It's a waste of your money. Because your soil already contains those nutrients. If you're not getting the nutrients into a plant-available form, how has nature been converting the mineral forms of nutrients from that crystalline structure in your sands, your silts, and clays? How has nature been converting that into a plant-available form for the last 4 billion years on this planet? This has been happening. What is it that does that? Well, guess what? You're looking at them right there. The plant is telling the specific species of fungus or bacteria that will go out and make the enzyme to pull phosphorus out of your rocks, out of your sands, your silts, your clays, your organic matter. It will choose to grow those bacteria that will pull the nitrogen out of that form. How much nitrogen does your soil actually have present in the crystalline form? And the only way to get that crystalline, that material that's in the crystalline form is to have the bacteria and the fungi making these enzymes and organic acids to convert it into, they convert it into bacterial biomass. They convert it into fungal biomass. Those nutrients aren't available to your plant. So this is not a one-step process. These are your holders. This is your pantry for your plant. The plant is telling these guys to make the enzymes to pull those nutrients into an organic form in the bodies of the bacteria and fungi. But now, how are you going to get the nutrients that are present in the bacteria and fungi into a plant-available form for the plant roots to take up? You got to have these guys. Life, nature is so clever. This is just so elegant, the way this naturally happens. You've got to have the predators of the bacteria. So the good guy protozoa, the amoebae, the flagellates, they are going to be eating the bacteria. And because the nutrient concentrations in the bacteria is so much greater than what's present in the protozoa, that there will always be excess nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium, iron, zinc, cobalt, anything you want to talk about. For this bacterium to be alive, it has to have all of those nutrients within itself. And when it's eaten by the predators, that's way too high a concentration. So this guy is going to spit those nutrients out as a plant-available form. And where is most of this interaction going on in your soils? Well, remember, it's your plant that's feeding these guys, so this elevated ability to grab nutrients out of your sand, soil, clays, rocks, pebbles, and everything else organic matter, it's right around the root system. So of course, where are the predators going to be attracted to? The root system. So where is most of the plant available nutrients being made available? Right around the root system. Isn't this clever? Isn't it just, wow, that's how it works. You've been watching this your whole life. You need to understand exactly what's going on so you can manage your system appropriately so you don't have to work so hard. How would you like to work less? So if these organisms in the soil are taking care of your disease problems, they're taking care of your fertility problems, they're making certain the roots go down deep so every drop of water infiltrates and is held in your soil, could you just like go fishing for the summer? After you plant in your field, please don't destroy the biology in your soil when you do that. Then you can go fishing until you come back and, well, okay, maybe you like to do other things like riding horses or whatever, but you're free to do what you want until you come back and harvest. You have to manage this resource properly, because if we get this biology back into the soil, we don't have to worry about fertility. We don't have to worry about weeds. We don't have to worry about pests. The toxic chemicals are not part of the solution anymore, unless every once in a while Mother Nature sends a nasty storm, a tornado or two wanders in. Okay, your soil is now over in the next province. You're going to have to, we need those fungicides. We need the inorganic fertilizers for those rare conditions where some bizarre event has occurred. So it's not like we outlaw those chemicals. You use them as necessary. So let's see, let's keep going. We need the nematodes. There are fungal-feeding nematodes that do the same thing for fungi, release those excess nutrients from the nematode's point of view, and feed your plant. It's kind of like the pizza delivery system of the soil. Your plant calls up the pizza parlor and says, hey, pizza parlor, I want cheese pizza because I'm lacking those nutrients. I need that cheese pizza. So the bacteria and fungi get to work. They've got their enzymes going, and they're going to make that cheese pizza. And then along comes the pizza delivery guys, and they are going to deliver the nutrients to the surface of the root. And your plant just goes, well, what took you so long, guys? I ordered my pizza 10 seconds ago. How come you weren't here faster? Because that's the time scale at which this works. It is so fast, it is immediate. If we have the biology in the soil, that plant is going to get every nutrient it needs. Because remember, it's not just nitrogen. It's not just sulfur. It's not just phosphorus. It's all the nutrients being delivered to your plant in a form your plant can utilize and take up and instantly be not stressed. Your plant should never be stressed for nutrients. Do you need 800 parts per million nitrogen in your soil for your plant to be healthy? That's way too much. Soluble nitrogen should not be present in more than 1 to 3 parts per million at any instant in time. So this system is constantly cycling, constantly supplying to your plant all the nutrients it requires. Your plant takes up what it needs. Your plant is never stressed. We don't need, and it's detrimental to be putting into the soil these massively high concentrations of soluble inorganic nutrients. Because how many of those soluble inorganic nutrients end up in your plant when we're putting on massive doses once a year? 80% of those soluble inorganic nutrients are going to be washed out of your soil. And where are they going? Into your water. We're destroying water quality. That's what's going to take people out. Long before we become food limited, lack of clean water. Do we already have that problem? State of Iowa in the United States. You cannot use any of the groundwater in that state because it is so contaminated with pesticides that it's toxic. You can't go to your own well and drink your own water. because of the damage that we've done with those toxic chemicals. How do we clean that up? Guess what? There we are. We have to start understanding this biology. It's another tool in your toolbox. Understand how to manage it, how to work this, and okay, in those situations where the biology's been wiped out, then you are going to be stuck using the toxic chemicals, but understand what the fungicides do. Understand what the bactericides do, and the herbicides, and the... What does that end of all of those words mean? I-C-I-D-E, aside. What does that mean? To kill. And they kill things, but they kill way more than their target organism. You can't kill just blight or rust with any toxic chemical. So sometimes you have to use them, but then as soon as you use those chemicals, those toxic chemicals, you have to put this biology back into your soil. How are you going to get these organisms back into your soil? Where's your inoculum? How many bacterial species do you need per teaspoon of soil, per gram of soil? 75,000 species of bacteria are present in a healthy soil. Go out and figure out how many bacteria you've got in your soil right now. How many species? If you've been putting on toxic chemicals, if you've been tilling, you have nowhere near that. How many species of fungi should be in a healthy soil? So we're looking at 25,000 of them. We need 10,000 protozoa split amongst about 1,000 different species per gram of your soil. We need at least 25 different nematodes. We want bacterial feeders, we want fungal feeders, we want the predatory nematodes. What's the kind of nematode you do not want? Root feeders. I'm certain that some of you have problems, and probably pretty astounding problems with root-feeding nematodes. Mother Nature is sending you a message. When you see disease, when you see a lack of fertility, when you see problems happening with your crops, nature is sending you a message. She's trying to make you pay attention and understand that you're farming with dirt, not soil. You need to get this biology back into your soil, make sure it's functioning. What is it that selects against your root feeders? You put out a nematicide. How effective is that nematicide? Because it only affects, maybe the top 6 inches of your soil. How far down into your soil do the root-feeding nematodes go in the winter? 12 feet. They're down there. So you use a nematicide in the fall or in the spring, it's not taking care of the problem. As soon as your plant starts growing, those exudates coming out, and there's no bacteria and no fungi, because most of our nematicides are better at killing bacteria and fungi than they are at killing nematodes. Nobody to use those exudates. And so those root feeders are going, oh, things are happening up there. Let's go back up and have lunch. And now they're going to start attacking your root systems. Get the rest of the food web in here, and these guys will never detect. The root-feeding nematodes will never detect that there are roots in the soil. My husband is a nematologist. So you can imagine our dinner time conversations. All of your organisms ate all of my organisms. What are you going to do about that? So nematodes, some of them eat fungi, some of them eat bacteria. And so we want these good guys. We want the predators. Here's one of your major defenses against those root-feeding nematodes. Predatory nematodes in general, I'm generalizing here, but in general, their favorite food are the root-feeding nematodes. You want to get rid of these, like, next week, want your root-feeding nematodes gone? You got to get the predatory nematodes into the system. These are guided missiles. They go directly to, because, that's like apple pie to a predatory nematode. Well, oh, yeah, they're having a grand old time. Take them out in a very short period of time. So we've got these predators eating those guys. which releases plant-available nutrients. What if you have too many of these guys? What happens when their population gets really high? Well, something has to eat them to keep their populations in balance with the populations of their prey species. So what else do we have to have in this food web to make sure we don't get too many of them? We got to have the things that eat them. So we have to have enough of these to keep them in balance so we don't shut down the whole system. Well, what if you get too many of these guys? And they eat all of your predators, of your bacteria and fungi. Sorry, no more nutrient cycling going on. So we have to have something that keeps them in balance. Okay, there we go, there we go. What's the top of the food web? Us. Our job is to manage this properly. So you don't have to work so hard. So our plants will have all the nutrition in them that they require. So we need to get this reestablished. And I could spend a couple more hours on this picture, but let's keep going. This is just a summary of what I just talked about, all the benefits that we can get. So we suppress disease. You don't need those toxic chemicals unless you wipe out your biology. You don't need those pesticides. We're going to keep all the nutrients in the system. No more runoff, no more erosion. Glue your soil together so it stays in place. When water moves into the system, it doesn't run across the surface, it infiltrates. And you store that water in your soil so your roots can get to it in a dry summer period. We make nutrients available because the bacteria and fungi, which are doing the retaining, are eaten by their predators, and that makes those nutrients available to the plant right at the root zone, not out 10 feet away from your plant. What good are soluble nutrients going to do for your plant when it's 10 feet away from a root system? Yeah, that's going to end up in your drinking water. And you're going to have to pay money to clean it up, or ultimately you can't drink that water. We decompose any toxins that might be in your soil. We're doing a lot of work in the desert Southwest to deal with uranium problems, to deal with cobalt problems, with all kinds of toxic materials that people have dumped into the soil. If I had enough time to talk, and whoever's keeping track of time, you got to stop me when it's, give me 5 minutes notice if you would, because otherwise I'll just keep talking all day. We had a project in Texas where they asked us to take a parking lot in the city of Dallas and convert it into native grassland so they could run some cows on that property. Yeah, right, downtown Dallas. So how are you going to do that? When we brought equipment in, it took 6,000 pounds per square inch of pressure to break through the top of that old parking lot and start pulling that, well, it wasn't soil, was it? And I wouldn't even call it dirt. So because we mixed the proper biology and organic matter, we had good compost with all the right organisms in it, all that good organic matter. Within 1 year, we had converted that from a parking lot into a native Texas prairie. The Lady Bird Johnson Wildflower Institute in Austin, Texas, came out and looked at that prairie, and they gave us an A++ rating. There were no weeds. All of the native grasses were growing. We had shrubs. We had the native trees in the system, and that was in a drought year in Texas. We did not have any rain at all from April until the end of September, and yet we managed to grow that prairie from bare soil to full, lush production through that drought, because you have to sequester all of the dew that falls. Do you have dew in this part of this world? Quite a bit. That's a massive amount of water. You don't want it evaporating off into the heavens. You do not want it running off the surface of the soil. You want every drop of that to infiltrate. You must have structure. You have to have the organisms to build that structure for you. So, talked about this, so I think I can go right on. A little bit of my background. So, yeah, I've been a lot of places on the planet. I've worked on every continent except Greenland. I've never been to Greenland. I'm not sure I want to go. So I know about biology and what helps grow crops. So methodology is a huge part of all of this. A lot of times when we deal with the chemical world, they will say, we have data that shows that fungicides or nematicides or any of these pesticide materials don't affect any of the beneficial organisms in the soil. Oh, really? What's your method? Using a particular medium. The other half, they'll take from the pesticide part, they'll plate it on exactly the same medium. And then what they observe is that there's no difference in the number of bacterial species or the number of bacteria based on what they're seeing on this plate count. They're pulling the wool over your eyes, because this is a completely inappropriate method for measuring these sorts of things. Plate counts were developed for human medicine. When we're trying to grow a particular pathogen, that's what plate counts are for. Because there's only 2 kinds of foods in this particular medium. How many different kinds of organic matter are present in a soil? Hundreds of thousands of different kinds of food resources. And you're trying to grow all of those bacteria and fungi. Each one of those species requires a different mix of food resources, of temperatures, of moistures, and they only grow under those very specific conditions. And you're going to try to pretend to me that this is going to give you a total bacterial count or a total fungal count? There's only 2 foods in here. This is tryptone, soy, agar. Tryptone, one particular kind of amino acid. Soy, how many proteins are present in soy? How many different kinds of food resources are present in soybean? Take a soybean, boil it to death in water, make a soybean soup. You dry it down, and then you can make a medium like this. There's 12 different sugars and proteins in soy. So you are not providing all of the nutrients to grow all of the bacteria and fungi in the soil. So can I pull the wool over your eyes? There's no difference where we just had water or we put in a pesticide. So see, this proves my pesticide does not harm any non-target organism. Yeah. What would you have to do to really be able to make that statement with a plate count approach? Whereas I take exactly the same sample. This is work from some oyster work that I was doing in Galveston Bay for my master's degree. Plated 1 drop of water, or 1 drop of the oyster's digestive system on this Petri dish. And you've only got 2 different species that's coming up on this plate. So most of the bacteria in there are, if the colony looks the same, it is the same species. There's a different species right there. So there's 2 species of bacteria in an oyster's digestive system. Looking with a microscope, we're looking at 1/1600th of a drop of the water, or the drop of the oyster's digestive system. That the drop is what went on there. Another drop where we're looking at 1/1600th of that drop. 400x total magnification. In this field, see all those little bacteria, all those little dots? There's a microaggregate. And so we have about 500 bacteria in that little aggregate right there. And all these other bacteria, and you can see where we have some short little rods, fat little rods, skinny little rods, long rods, short rods. When you look through all of the things in that drop of water, you have over 1,000 bacteria in this 1/1600th of a drop of water. There are 12 different species in that field. And we're seeing protozoa, which you are not picking up at all on this Petri dish. So which one is telling you truth? And now you understand why this is completely inappropriate. So don't fall for people using this kind of methodology. And we're excellent. We know how to grow E. coli because it's a human pathogen, and we need to know. So plate counts are fine for Pasteurella, for Salmonella, for Shigella, for Legionnaire's disease, for all of the human pathogens. That's the way to figure out how many bad guys of a particular species are present, but totally inappropriate for growing all the bacteria or fungi or protozoa that might be in your soil. There are other assays that people like to use that, again, pull the wool over your eyes. Please realize that when bacteria grow in soil, they release 60% of the carbon in their food as CO2. But fungi only release 20% of that carbon as CO2. So if you're using carbon dioxide evolution methods, you are being handed some mythology again. Because it's very important to know, is that mostly bacteria that are present in that material? Do you have bacteria and fungi, or do you have just fungi or predators? Lots of things you would have to know to be able to interpret those things correctly. So we've covered all of this, so I'm going to just keep going. I want to get on to not just the difference between soil and dirt. Let's cover this. I've got a few more minutes. How much longer do I have? 15 minutes. Okay, we can get through this. Remember where I was talking about the difference between soil and dirt. Dirt is where you have just the mineral component of your soil. So what's soil then? This is the three components of soil. If all you have is the sand, the silt, the clay, the rocks, the pebbles, that's dirt. In order for it to be soil, you must have organic matter because you need the food to feed the microorganisms. And by definition, soil must be aerobic. It cannot be soil if it's compacted, if it's gone anaerobic. If your soil goes anaerobic, you're losing all your nitrogen. Your soluble nitrogen is going to be lost as ammonia gas. You're going to lose your sulfur, your soluble sulfur, as hydrogen sulfide. You're going to lose your phosphorus as phosphine gas. If you go anaerobic, the pH is going to drop below 5.5, and that's going to start killing your plant because acidic pH is going to start solubilizing your roots. Very toxic material, only produced under anaerobic conditions. Formaldehyde formation in your soil only occurs in anaerobic conditions. Alcohol production in your soil, which will, again, solubilize your root systems. What do I mean when I say solubilize your root systems? Let me put that into average, everyday language. It turns your root systems into mush. And it's really hard for a root to take up nutrients and feed it to the above ground part of the plant when it's basically been turned into mush. So we don't want things to go anaerobic. By definition, Hans Jenny, one of the fathers of soil science anyway, defined it as it's got to be aerobic. As soon as you start losing oxygen, it's not soil, it's dirt. Because pretty much all you have left are the pathogens and the pests, and of course, the mineral component in your soil. What are the minerals? How many mineral nutrients? And people often just, they think this is soil. No. This is the texture of your soil. The percent clay, the percent silt, the percent sand. I'm sure you've all seen this. And so what kind of soil do you have? I can take a pure sand soil. I've done this to USDA extension surface guys quite a few times. We'll take a soil that's total sand or total silt, really bad stuff. And the extension guy will come out and say, oh, you've got really poor soil. You have no nitrogen. You have no phosphorus. Really? Really? What's in the crystalline structure of that sand, silt, and clay? Let's look at what's actually present in terms of nutrients in our sands, our silts, and our clays. So in this table, taken from Sparks' work back in 2013, he went and he measured, it's got to be about 3 million soil samples from all over the world. And you have the average value of all these nutrients, and he did all the rest on the periodic table. I've only put the top 20 in here. So this is the average value of all of these nutrients averaged over all soils on this planet. Now, I'm not talking marine mud. I'm not talking about stuff at the bottom of the ocean. Those are not soils, are they? So, soil. How much nitrogen is present in your soil? On average, in general, 2,000 parts per million, 2,000 kilograms per hectare of nitrogen present in your soil, in the mineral material. Why would you ever put on a nitrogen fertilizer? Oh, your soil's poor, doesn't have enough phosphorus. Really? Where's our phosphorus? 800 kilograms per hectare? Why would you need more phosphorus? What if you don't have the biology to convert these into the plant available form? But the nutrients are in your soils. Until the day you run out of sand, silt, clay, rocks, pebbles, gravel, parent material, bedrock, you should never have to put an inorganic fertilizer on your soil. You're wasting your money if you do so. So how many of you lack rocks in your soil? So how long before you have to start worrying that you're going to run out of nutrients? Well, you have to get back into the soil as your organisms. And the easiest way to do that is make decent compost. If you make aerobic compost, we can kill the pathogens and the pests and the parasites through the use of worms or through the use of temperature. We can take out the bad guys and leave just the good guys. We have to make certain that all of that organic matter stays aerobic. So if you go to your municipal composting yard, I'm sorry, that's not compost. That's reduced waste. Call it what it is. It's gone anaerobic, most likely. How do you tell? Color. If it's black, it's not compost. It should be a rich, dark brown color, about 70% cocoa. You want a standard that you can take with you out to compare whether this is good compost or not? Go buy a chocolate bar, 70% cocoa chocolate bar at the grocery store and bring it with you. And you go out to the composting facility, the municipal compost yard, where they're actually making reduced waste, not compost, and you compare the color. Dig into the pile and compare the color of the chocolate bar. So after you spend a whole week going around all of the places here, this part of Canada, because I've had people do this, they've gone to all of the places that sell compost, and there was not a single place that was actually making compost. It was all reduced waste. Now, that might have changed in the last couple of years because it's been a while since I've been in this part of the country, but you need somebody making good compost. It's not that hard. It takes about 21 days to make good compost if you're managing it properly. So your soils, there's no nutrients lacking. I know sometimes people will, oh, look, your molybdenum is really low. Oh, yeah, but this is milligrams per kilogram or kilograms per hectare, about the same thing. If you just do the conversion, how much molybdenum does your plant actually take up in the course of its lifetime? It takes up about 2 parts per billion. So if you've got 1.2 parts per million, you've got, only 1,000 years of nutrient in that particular gram of soil. If you wait, those bacteria and fungi continue to decompose your rocks, your pebbles, your boulders, your bedrock every single second of every day through the whole entire year. Microorganisms are decomposing your rocks, your pebbles, your gravel, your parent material, so your soil is being replenished every second of every day. So until the day you run out of these things, you don't have to worry about fertility, as long as you have the organisms that will convert these nutrients into plant-available forms. You want to stop working so hard? Put your organisms to work for you. They are free. All you have to do is remember to feed them. And the way you feed them is to plant a plant and let the plant do the work for you. The plant's very good at knowing what it's lacking. How many of you, now think about if you've got a greenhouse or something, how many of you would know right this second, right now, today, what's the mineral nutrient that's preventing your plant from being able to grow in the most healthy fashion? How many of you know? It's cadmium today at this site. Oh, wait a minute, now it's manganese. And now it's something else, and now it's something else, and now it's not just one nutrient that your plant requires, it's all of them. So how do you provide all of the nutrients so your plant can take up what it needs every second of every day? Get the biology working for you, and then you don't have to worry about it. So why is it we go out and we put inorganic fertilizers on our soils and we see the green up of the plant? Yeah, because that plant is stressed. It's not getting the nutrients. It's not being presented. The pizza delivery system is not working in your soil. That's the only way when you put on an inorganic fertilizer that you see a response. Mother Nature is trying to send you a message. There's something wrong with biology in your soil. Fix it. Because we don't do a very good job of providing that plant precisely what it needs every second of every day. If all you're putting on is nitrogen fertilizer or phosphate fertilizers or whatever one or two varieties of fertilizers you're putting on, are you putting on everything that your plant requires? So why do we have nutrition problems? The plant doesn't have the nutrition in it. If you don't have the nutrition that you require, are you more susceptible to diseases and problems? Same thing with your plant. If it's stressed, it's not going to be able to ward off the diseases. So keep going because I'm almost out of time. When nutrients are removed from your field, so you grow a crop of soybean or you grow a crop of wheat or whatever it is you're growing, and you harvest that, isn't that a whole bunch of your nutrients being taken away? Don't you have to replace those nutrients? They're already in your soil. And until the day you run out of sand, silt, clay, rocks, pebbles, organic matter in your soil, you don't have to worry how much you're exporting from your field because it's already there. Your rocks, your pebbles, your sands, your silt, they're going to be replenished because you got that wonderful bedrock down there where the microorganisms every second of every day are releasing more nutrients, and your soil mineral component is rebuilt every second of every day. So when are you going to actually end up running out of sand, silt, clay, rocks, and pebbles? I don't think you have to worry. Because if we actually don't have any bedrock left, we don't have a planet. So I think we're safe. So we don't have to worry about that. Think about old-growth forests where nobody has ever put out any inorganic fertilizer at all. And yet they have more plant production on an annual basis. Oh, well, nobody's harvesting the forest. Yes, they are. That tree, how much nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium, all of the nutrients are being sequestered and put away into the trunk of that tree. Every year there's that increment of growth that every single tree in that forest is taking nutrients from the soil and sequestering it. It's not going back into the nutrient cycling system. Are those trees dying from lack of fertility? So if old-growth forests can be there growing for 1,000 years or longer, think about some of the redwoods in California that were young trees at the time that Jesus was born. And they're not dying from a lack of fertility. They're not dying from pests attacking them. They're not being attacked by diseases. So we have to rethink some of that mythology that we've been taught. So I have 2 minutes left to go. Is that what? Okay. So should we do a couple of questions?
If you're done, you can, yeah.
I'm not done. I could keep going. If you're trying to grow corn, and that's the biology that we need. We need 600 micrograms of bacterial biomass, and we need 600 micrograms of fungal biomass. And I can guarantee you right now, this is what you're missing. You probably have the bacteria in your soil. What you're missing, say you're trying to grow soybean. Soybean is going to be a little bit more on the fungal side. So you might need 700 micrograms of fungi, 600 micrograms of bacteria. And I can guarantee you that probably what you're missing is that fungal component. You're missing the protozoa. You're missing the good guy nematodes. How can you possibly control your root-feeding nematodes if you don't have the beneficial nematodes in your system? You're forced to use the toxic chemicals. And that means you're out there driving, well, some of you like to drive around on your fields, okay?
Thank you very much. I think
Okay.
we're going to wrap it up now. Let's show our appreciation, please. We're going to switch to the second